que eles têm lá alguns laboratórios, o nome dele é PRL, né? Então, quando eu vi, eu já lembrei de Physical Review Letters, né? Que para gente físico, né? Mas o acrônimo é Product Realization Lab, né? Então, são laboratórios de realização de produtos, né? E são 3 na universidade, porque o campus é relativamente grande, então tinham 3 centros, né? Com mais ou menos verticalizados. Por exemplo, impressora 3D e prensa de moldes, outro mais marcenaria, o outro até fundição, né? Todos com a eletrônica. E era assim, basicamente, se você fosse aluno, você pagava $100 por ano, né? Que eram $100 muito bem gastos, e aí você entrava no ecossistema dos PRLs, né? E era mais ou menos assim, um laboratório onde os próprios usuários mantinham um laboratório. Então, se você entrava, né, e queria usar, por exemplo, impressora 3D, que eu acabei usando bastante, obviamente eu não tinha o conhecimento de operar, então algum usuário mais antigo me treinava, né? E aí eu desenvolvia lá meu produto, fazia algumas, né, imprimia, interagia com eles e tal. E a ideia era essa: se você tem uma ideia, né? Poxa, tive uma ideia de fazer um suporte de celular, né? Mas não tenho vias de começar a fazer o meu minimal viable prototype, né? O prototipo minimamente viável, né? Para eu testá-lo, né? E dá para os outros testar, né? Aquela ideia que eles têm lá de Lean Startup, né? Que é aquela startup enxuta, né? Rápida e tal. Então você ia lá, né? Alguém te treinava, você imprimia, interagia, conversava com algumas pessoas que estavam com o mesmo intuito que você, né? Dali saíam novas ideias, novas interações. Eles organizavam muito noites de pizzadas, assim, onde pessoas que já saíram de Stanford, até do próprio PRL, né, e já tinham empresas que estavam num estágio mais avançado, iam lá contar os cases, né, deles, e você interagia. Numa dessas, eu interagi com a época, o CEO daquele Snapchat, né, que era basicamente uma mídia social muito forte lá nos Estados Unidos. Cheguei a ver, até engraçado, mas uma palestra da Elizabeth Holmes, da Theranos.
Aquela que está na cadeia, né?
Exatamente, exato.
Eu
Eu fui para lá em
acompanhei
2014.
essa história.
Exato, e ela estava no auge, assim, era 2015, logo depois deu o downfall dela, né?
Eu morei 21 anos nos Estados Unidos, fiz 9 anos de pós-doc e 12 eu era professor em uma universidade em Michigan.
Que legal.
E eu acompanhei toda essa história dela aí desde o começo.
Pois é. Foi assim, foi uma surpresa, né? Porque
um negócio meio trágico, né?
Meio trágico, e muito trágico.
Até o Henry Kissinger estava envolvido nessa história.
Pois é. A CVS entrou com força. A CVS comprou a ideia, tudo. Mas o pessoal lá tratava ela como o next Steve Jobs, né? O pessoal colocava ela num pedestal também. E acho que criou ali uma percepção talvez até equivocada, e ela tinha uma oratória, assim, convincente, sabe? Eu vi a palestra dela, ela tinha uma voz bem empoderada, assim, né? Uma voz... Ela falava bem, né? Mas
os americanos falam bem. Né?
Exato.
O americano médio fala muito bem, eles são treinados na escola.
Eles articulam, né?
Exatamente. Meus filhos fizeram a educação toda lá, né? E deu para sacar isso daí. Eles são chamados sempre na frente da classe para falar, para expor, não sei o quê, tem todo um...
Isso é importantíssimo, né?
Você tem que aprender a falar para um público.
Pois é. De fato, eu percebia muito isso, né? Uma eloquência muito apurada, né?
E uma autoconfiança.
Exato.
Porque esse negócio te dá autoconfiança.
Sem dúvida.
Eu posso enrolar qualquer um. Ou posso contar a história que eu preciso contar. Pode ser enrolação, pode ser o cara está apresentando uma verdade, mas de uma maneira muito boa, porque eles são eloquentes, né?
Exato. Não, sem dúvida. Mas foi uma época boa. Vamos lá.
So let's move to English. So
Sure.
let's start. I am already recording. I always forget to start recording. So today we have with us Professor Gregório Faria. He got his bachelor's, master's, and PhD at São Carlos, USP São Carlos. During his PhD, he spent some time at TU Darmstadt in Germany. And then he did a postdoc there in TU Darmstadt, a short postdoc after he was hired at, between hiring and assuming his position in São Carlos, and a longer postdoc that he was telling us about at the beginning of this conversation at Stanford University. And since 2013, he is at USP São Carlos. And today he's going to talk to us. The title is The Ballet of Ions and Electrons, Fundamentals and Applications of Organic Mixed Conductors. Professor Faria, it's a pleasure to have you here. Thank you very much for accepting our invitation, and the microphone is yours.
Thank you very much, Professor George. Thanks for having me. Here is really a pleasure to be giving this talk to you guys. I have good friends at University of Federal of Uberlândia, Professor Marleta, that probably is here, or if not, some of his students are. And I also, during my master and PhD, I interacted with Professor Roberto Tozzoni, that might be also around. So it's a really great pleasure to be giving this colloquium for all of you. So basically, I'm going to talk about mostly science, and those are, results that I've been, or a vertical of my research that I've been interested on recently, and they are actually gravitating towards this organic mixed conductors. So I'm going to talk a little bit of fundamentals and some interesting applications that we have recently published, and that might interest you as well. So to start with, I guess I have to touch a little bit on these questions. What is mixed conductors? And of course, answer why organic, right? So those are materials that basically support electronic and ionic conduction with equal efficiency. So you can have both conductivities, ionic and electronics. And the organic here comes from because mostly the active material are polymers, or it can be small molecules as well, but I'm going to focus mostly on polymers, and especially conjugated polymers, which are, the class of polymers that provides the ability of conductivity and semiconductivity. And the reason we are interested in mixed conductors, there is, of course, a lot of reasons, but especially in my case, it's because we as mankind, our technology, we are very good in constructing electronics that deals with flux of electrons. So all our devices, processors, and equipments, they are all very good for measuring electron fluxes. However, if you look into biology and our body and all the biological species, they mostly communicate through ionic fluxes. So the case of, for instance, a neuron, the way neurons sort of communicate is open and closing, some sort of ionic channels that, splash ions into the intercellular spaces, polarizing those spaces, and then the next neuron sort of feels that polarization and send the message across, that particular string. So if you want to communicate or interface or mimic biology, it's really very important to be able to measure or to transduce or to sense those ionic fluxes. And we do it through the electronic portion of our technology. So having this mixed conductor is very good because you can bridge these two different worlds, right? And if you think about polymers and organic molecules and polymeric materials, they are mostly insulating materials. They protect us from, from the electricity, like in this small little picture here. But interestingly, we have a class of materials known as conjugated polymers that present semiconducting properties. And in some cases, because of the doping and de-doping process, you can also have a very high conductivity as well in these materials, make them very attractive to the electronic portion of our technology. And just to quickly overview the differences between conductors, insulators, and semiconductors, let's start with conductors. So we basically, in terms of the energy diagram, we have two types of conductors, those that have high band gap. However, the valence or conduction band are partially filled, like in this illustration here. And therefore, electrons with little absorption of energy, they can actually move across empty states, and then the conductivity are very high. The other part of conductors are basically those with very small or zero band gap, in where you have a valence band completely filled, but therefore, because of the very small band gap, electrons can easily hop into the conduction band and therefore get a very high conductivity. So those are the conductors. If you move to insulator have a very high band gap, filled valence band, of course, and therefore, because of this very high band gap, it's not very easy to remove electrons from the valence band and promote it to the conduction band, and therefore conductivity is very limited on these materials. And here's where most of the polymer materials rely on. The aliphatic polymers, they have energy band gaps of around 6 to 10 electron volts. So it's really difficult for you with the surrounding energy to be able to promote electrons from the valence band to the conduction band without breaking the cohesion of the molecule. And in between both those two big worlds of insulating and conductors, you have the semiconductor, where it's basically similar to insulator. However, the energy gap is smaller and rely on the energy that normally we have in our surroundings, which is between 1 to 3 electron volts. So every material that has a band gap, a filled valence band, and a band gap of around 1 to 3 electron volts, they behave mostly as a semiconductor. And the way actually polymer sort of becomes a semiconductor is actually, it can be very complicated, but I'm gonna try to make it simple. And I love to start with this simple idea of having two hydrogen molecules separated apart, and you start bringing them together. So when you do this, if they are, fall apart, they are very far apart, you have the wave function of these two twin atoms very similarly. And as you bring them together, those wave functions, they start interfering with each other. And they can interfere in a constructive way or in a destructive way, depending on the spin of the electrons that are actually within those atoms. And by interfering either constructive or destructive, you can actually create two different types of molecular orbital. And we can think about not the wave function themselves, but also, but especially the square of the wave function that gives mostly, the probability of finding an electron, around the nuclei of those atoms. And when they interfere constructively, you see that mostly of the electrons, the probability of finding electrons are in between the nuclei of these atoms. And therefore, the constructive molecular orbitals sort of creates the cohesion of the molecule that glue the atoms up together, and therefore forms the molecule. And the destructive way, if you sum them up and of course take the square moduli, you see that there is a node in between those two nuclei, and the probability of finding an electron in between those atoms are actually almost zero or negligible. And therefore, this one doesn't really add to the cohesion of the molecule. And for those two states or molecular orbital, we call bounding molecular orbital that actually binds the atom together, and anti-bounding molecular orbital that actually breaks them apart. And those names comes from basically, the fundamentum of the quantum mechanics and the probability of finding an electron within the nuclei. And interestingly, if you take those atoms far apart, hydrogen A and B, and they are very far apart, and because they are very similar atoms, they are equal atoms, they have similar energy, and as you bring them together and they start forming a molecule, because of the exclusion principle of Pauli, those energy that were degenerated before, they start moving to different energy to, of course, obey the exclusion of Pauli principle. And therefore you start having this difference of energy between the molecular orbital, which is the one positive, which is the bounding molecular orbital, and the negative one, which is this one, which is the anti-bounding molecular orbital. And by this very simple idea, you start getting an idea of how these band gaps start to be formed. And in conjugated polymers, and by conjugated polymers, I'm referring to molecules that have alternating single and double bonds. So you have single, double, single, and etc. If you solve the tight bounding model to this type of structure, you realize that for those carbons, the band gap more or less rely between 1 and 3 electron volts, giving to this class of material the ability to sort of be a semiconductor. But when you talk about polymers, you don't talk exclusively about very defined energy states of the molecular orbital. We normally talk about bands, and the reason is when you start growing those polymeric materials, and by polymers, remember that poly is several, meros is a repeated unit. So you start putting a lot of this similar molecule all together like this. You start adding more and more carbons to that addition, to that sum of wavelength, of wave functions of different carbons. And because they have to always obey the principle of exclusion of Pauli, they are not always at the same level. They are a little bit apart from each other. And as you grow and grow and grow this molecule, more and more states, they have to sort of they have to sort of organize themselves among this two energy of the bounding and anti-bounding molecular orbital, starting forming a little fuzzy state, which we call like a band. And that's the reason most of the polymer, if you take the absorption of emission of those polymers, they are not monochromatic. They are actually very spread out in a wavelength of energy, is given this sort of principle that comes from basic molecule formation. And interestingly, you can model this in a very simple way, thinking about very basic quantum mechanics and the problem of particle in a box. And because here I have a very linear conjugated backbone, I can think that all this, orbital that actually conducts the electrons, they sort of overlap, and therefore an electron that is in this carbon here, mostly here, it's all delocalized throughout all this polymer chain. And because it's all delocalized, I can think of that electron as being, in this problem of particle in a box, where the box has a length of L. And by simply doing quantum mechanics, you can calculate the energy of this electron, and basically is inversely proportional to the square of the length of the well. And actually, people start looking into this basic chemistry / physics of this polymeric material. And there is a very interesting paper from 1997, it's a long time ago, but those folks here, I think they are from Santa Barbara, if I'm not mistaken. They took a traditional polymer, is a polyparaffiniline derivative, and they were able to, use the monomer, 1 unit, and some oligomers, 2 units, 3 units, all the way to 11 units. The beginning of a small little polymer. And here they are measuring, the maximum wavelength of absorption, which is related basically to the band gap of that particular polymer, or basically, the energy that a photon needs to have to be absorbed and promote an electron from the bounding molecular orbital to the anti-bounding one, which is basically the band gap. And therefore, this lambda max sort of resemble this energy and the energy of the band gap. And as you can see, if I have a very small molecule, it is a monomer, it's just 1 unit of this polymer, the wavelength is small and therefore the energy is high. And as I start growing this molecule, you can see that the wavelength increases, and therefore the energy decreases, mostly obeying this kind of law, very simple model of electron in a box, sort of, validating all the discussion that I have made so far. And polymers are not so simple as this picture that I'm actually trying to make. Polymers are very complicated materials, and I would wish that polymers would have this very straight and very well-behaved morphology or organization, but they actually have some sort of twist. And you can think about a rope. If you have a very long rope, they normally twist. They actually have very complicated organization. And this is very similar with polymer. They are very long, and so they start actually kinking and torsions of the backbone. And when you have torsions like this, where the molecules sort of twist 90 degrees mostly, one from the other, what you do basically, you break the conjugation of the superposition of the molecular orbital, and therefore the electron that was previously all delocalized throughout the entire conjugation length is now restricted to only part of this entire section. And again, thinking about the problem of particle in a box, now instead of having a problem with one electron being delocalized in a particle, in a box of length L, now I have an electron that is localized or delocalized in a box with L over 2. So now I start changing the energy. Again, the same molecule, but just because this particular molecule actually had a torsion, I just broke the conjugation, and now the electron feels an energy that is slightly different. So this is more or less what we call disorder, because when this polymer starts shaking and twisting and all distortions, you start adding disorder to the system. And all these disorders of the system, they actually tune slightly the energy that every electron in that particular molecule feels. And this also contributes to this fuzzy addition of several energy states that come from that simple model that I mentioned to you based on hydrogen molecules. So, although you can look at polymers in a very first principle view, which is very simple, they can easily become very complicated because you have to deal with morphology, you have to deal with conjugation length, you have to deal with the size of the molecule, basically what actually this article has actually shown to us, and therefore things can get out of hand very easily. But it's very important to know this basic because a lot of this will come during my discussion in the next slides. And here are a few examples of conjugated polymers. We have a bunch of those materials. Chemistry are very clever to synthesize those materials to, of course, harvest one or other property that somehow, undesired application needs. So you have a palette of enormous numbers of very complicated molecules indeed that are applied to either lighting emitting diodes, photovoltaics, transistors, and biosensor, and a lot of other type of materials and devices and applications, of course. Well, but this is a simple fundamental on this material, but I know everyone wants to see some sort of conductivity going on. And one of the most exciting example that we have, and I think successful example that we have in terms of devices in conjugated polymers are what we call the polymer lighting emitting diodes. So basically a LED, a lighting emitting diode, it's made of, you have an electrode. Here is normally an oxide that is transparent. You have your active layer, which is the lighting emitting polymer, and then a methyl electrode that sort of injects electron. So this one, the methyl electrodes inject electrons into the conduction band, while the ITO, which is the anode electrode, actually injects holes or extract electrons. And therefore, within the material, you have, a conduction on the conduction band of electrons, and in the valence band, a conduction of holes. And when they meet each other, they actually recombine and, sort of emit lights. So this is the very principle of lighting emitting diodes. And we have worked a lot on this. I've been working on other devices nowadays, but I think this is really interesting, and we can see this application nowadays even in the market. So this is, a very good proof of electron and hole conductivity within conjugated polymers. And here is a short example. It should be a video, but it's not. Oh yeah, there we go. So this is a flexible lighting emitting diode that we actually built in our devices 10 years ago. This is 2013. Time just go by very fast. It seems that was yesterday, but this is a yellow emitter that we managed to put together in a flexible display, and therefore you can bend and twist, and you see a very, very intense light of emission. And based on this example, we can make a lot of different progress. For instance, this one that actually is reported in this manuscript, for instance, we made use of a blend of different materials to build a light that or a lighting emitting diode that actually emits in the white color. So it's basically for application in lighting of rooms and etc. So there is a big interest on white emission. So, and it's not very easy to get those white emission because you have to combine several colors. And here we made possible, this white LED. Well, the technology is actually in the market nowadays. You can buy, TVs based on this technology. And here they call OLED, which is organic lighting emitting diode. And mostly because, instead of polymers, they also use organic molecules and sometimes even a mix of both. and you can make very small pixels, and therefore you can get very high resolution. Here is a 8K. I don't even know if our eyes can capture, or differentiate this one from 4K. But even though this is 8K, so apparently the pixels are even smaller, so the resolution is fantastic. And this is 70 inch, so it's a very big television. And this is another advantage of polymers per se. It's because you can process in solution, so you can print it, all this display very easily. Well, not very easily, but you can print it. And therefore the technology is nowadays available. So if you have 87, almost 88,000 Brazilian reais, you can go to Magazine Luiza and buy one for you. It's still very expensive, but the technology is there already for this type of applications. But well, this is a little bit of motivation, but let me now move a little bit to some interesting work that we have done recently. Well, not so recently. Time goes by very fast. So a few years ago, and one of the main problem with polymer when we talk about electron conductivity is what we call oxygen traps. Basically, oxygen has a very good electron affinity. We know this by basic chemistry, and we know that oxygen is all around us. That's the reason we are breathing, right? So a quarter of our atmosphere has oxygen. And when you build those devices, those applications, it's very likely that the polymer will interact with oxygen unless you seal it or you do some sort of protection, which is not very easy to do. And basically what happened when an oxygen sees a conjugated backbone, it sort of moves closer to the molecule and start interacting with it. And the reason is because you have a lot of electrons in those conjugated backbones because of the semiconducting property. And therefore, because of the electron affinity, the oxygen sort of traps or steals an electron from the conjugated molecule and p-dopes the molecule, because when you remove an electron from the polymer, which before was neutral, you leave a hole behind, and the vacancy of that electron is left behind. And therefore, there is a lot of problems with this charge transfer electron trap states. This is the big name that the community sort of calls it. And it's very difficult to sort of study this phenomena in order to, try to avoid it, right? So we decided to dig into this problem and try to, study this effect a bit in details. And for that, we have built a very simple diode here, sort of capacitors, where we have this ITO, which is the oxide, the transparent oxide. This material P3HD, which is a thiophene polymer. This is the structure here, the chemical structure. Very simple material, but very well known, and a very, very important protagonist in organic electronics. You can do transistors, you can actually build photovoltaics out of P3HD. It's a very well-known material. And as a cathode electrode, we actually put it silver. So this is the structure of our device. And here is sort of the band diagram of it. So the HOMO and LUMO, which is basically the valence band and the conduction band of P3HD, is around 3.1 for the LUMO and 5.1 for the HOMO. And because P3HD is initially neutral, the energy, the Fermi energy, or the Fermi level lies in the middle of the band gap, around 4.1 electron volts. And here are the work function of both silver and ITO. So this is basically, the energy diagram for this molecule. One of the big problem to measure current as a function of time in those sort of capacitor-like structure is the capacitive response. It's very complicated because we know that this is a capacitor, is electrode, and a dielectric. So this is basically a capacitor. And when you do any type of transient response, the capacitive response, which is basically the displacement current, will kick in. And sometimes it's very difficult to decouple or to differentiate from the displacement current and the conduction current for transient responses, because they sort of mix each other out, and you cannot sort of separate them and study the one you really are interested. So there is a technique that circumvent this phenomena, and it's basically applying a linear increase voltage across the sample through the electrodes. And by doing so, a linear increase in voltage, we know that the displacement current is the capacitance of the device times the derivative of voltage and time. And if you derive a linear increase voltage, this portion becomes a constant, which is basically the rate of growing this linear voltage. And because capacitance is a constant as well, the displacement current becomes just a constant current, which is actually illustrated by this blue box of currents. So you apply a linear increase in voltage, and then you measure this constant current. And therefore, if you have any free electrons or holes within your dielectric, and these, of course, are not displacement current, they are really conduction currents, that conduction current will actually appear on the top of this displacement constant current on this technique. And this is more or less what I illustrate here. So if I don't have any charge in the semiconductor, the technique would give me only this constant current, which is the displacement current. But if I do have charges in the semiconductor, it sort of appears as an extraction on the top of this curve. So I can separate the both currents. And what we did then, we measured devices which were unexposed to air, and therefore without any interaction with oxygen. And then after measuring those, we open up them to, to the atmosphere to start interacting with the oxygen. And we thought initially that, okay, unexposed devices, of course, will be neutral, have no charge within the semiconductor because, it hasn't seen any oxygen molecule, and therefore there is no p-doped phenomena or a hole left behind because of the charge transfer with the oxygen. So there is no charge here. The only thing I would actually measure would be the displacement current. And then we thought, as we start exposing the device to oxygen, this process start to happen, and then more and more holes will be added to the semiconductor, and then I would start extracting those current, and then I can study the kinetics of it, energy binding, and a lot of different phenomena that I can extract from it. So this was, the drawing board initially when we thought about those experiments. So we, of course, produced the devices. We were inside the glove box, protected 100% from oxygen, and then we measured. And this is a camera focusing on an oscilloscope, and this is times equal to 0, and by times equal to 0, I refer to the measurement of devices that are non-exposed. And interestingly, we saw an extraction here. Initially, we thought it would be completely empty, no current at all, because there is no charge in the polymer. We knew the polymer was neutral. So even not exposing the device, we would have, current extraction, which is this peak, this sort of Gaussian peak on the top of the displacement current. So we thought initially something would be contaminated. We exchanged the polymers, we redid everything and blah, blah, blah. And every time we would start this experiment, boom, the middle peak, or pK that we call this peak, pK, was there. So after sometimes we start, okay, there might be something else. So let's expose the device and see what happened. And this is actually in the film here. So as you start exposing the device, there is something very interesting happening. The peak that was originally for unexposed device start diminishing, and another one in the beginning of the ramp start decreasing. And you see that the rate they appear or disappear are mostly the same, suggesting that the effect of creating the new peak is actually diminishing or destructing the other peak. So we were very confused about this measurement, and we repeated, we exchanged the batch of material we thought was contaminated, etc., etc. But everything was always behaving like this. So okay, we thought, okay, let's get back to the drawing board and see what happened. And of course, we lost something, of course. If you look at the energy diagram here, and again, the energy level or the Fermi level of the electrons in the semiconductor is around 4.1 for a neutral material lying in the middle of the band gap. And you see that in this interface, the work function of the ITO, which is the oxide, is actually very big, is around 0.7 electron volts. And actually there is an equilibrium that has to be achieved because there is so differences in energy between these two work function or Fermi levels that in that particular interfaces, nature acts to sort of bend the energy to make that transition smoother. And, just to make an analogy, if you have two swimming pools, in two different levels, and you bring them together, the same idea is that, water from the upper swimming pool will move to the lower swimming pool in order to sort of adjust the level, because you would never see a swimming pool like this with different levels without, a well to hold, that particular amount of water on the upper swimming pool. So this is basically the same phenomena. So what happened when you build the device, you start having this band bending just so, that particular smooth of the interface would happen. And the way nature deals with this is actually exchange charges. So the ITO injects a lot of charge and creates a small little reservoir of charges in the interface of the P3HT with the ITO, and bends a little bit the HOMO of the ITO in that particular portion, create, a reservoir of charge that is proportional to the differences of energy between the Fermi level of P3HT and, of course, the work function of ITO. The same would actually happen in the silver, but because this gap is smaller, of course, the reservoir here is not as big as the one in the ITO, and therefore the ITO reservoir sort of dominates the process. Oh, oops. And then we sort of understand the nature of the pK, that peak which is related to the non-exposed device. That extraction of charges is basically extracting those charges that are actually at the interface between the ITO and P3HT to sort of work out the energy break at that interface. And you see that the extraction happens in the middle of the linear increasing voltage, meaning I need higher voltage to extract it, because those charges are very bind to this particular system because they are there to maintain equilibrium. So it's not easy to pull them out. And that's the reason, that particular charge is extracted at higher voltages. Remember that this is time, the x direction is time, but I have the linear increasing voltage also evolving in time, and therefore, this is for higher voltages as well. I have to run a little bit because I'm looking at my clock here and I see that time is ticking, but I'm gonna push a little bit. So this is more or less summarizing the results. So no exposed, only pK, and as I expose as a function of time, pKa sort of diminish, and peak B, which is in the beginning of the extraction, start appearing. And we can do a lot of things. We can integrate this curve here to calculate the amount of charge, and we did this over time, and therefore we can sort of create or recreate the disappearance kinetics of peak A and the appearance kinetic of peak B, and both actually have times appearing and disappearing times very similar, which means that the reason that peak A is actually appearing is the reason that peak B is appearing is the reason that peak A is disappearing. So it's a connected phenomena. So to explain why that is happening is actually quite simple. And the reason is, well, now we understand that for unexposed device, I have this big reservoir at the interface. And as I start exposing my devices to the oxygen, remember that I create this oxygen trap, and then I start p-doping the p-TRAT. And by start p-doping the p-TRAT, I add positive charge holes into the material. And when I do so, what I'm doing in terms of the energy level, I'm pulling the energy level towards the HOMO of the material. So the energy level that before neutral was 4.1 electron volt, now is moving towards the HOMO of the material. And by moving towards the HOMO of the material, I sort of diminish the energy gap between the Fermi level of the neutral p-TRAT and the work function of ITO. And therefore, I diminish the amount of charges that needs to be added to the interface to sort of bend and get the interface into equilibrium. And that's the reason pKa diminish, because the Fermi level is sort of moving down towards the HOMO. So as times go by, you add more and more P doping to the p-TRAT, more the Fermi level moves toward the HOMO, smaller is the reservoir of charge at the interface, up to the time that basically that reservoir disappear, which is basically what we saw in the results. So we managed to explain all these intricacies, and moreover, we did more experiments. I'm not going into details because I'm gonna move a little bit to ionic conduction, but we were able to measure kinetics of those processes to show how fast they occur, energy binding, and etc. And we sort of pave a new road to characterize those reservoir of charges at the interface, which before were not able to be achieved, and as well as those p-doping due to oxygen effect. Okay, let's move a little faster. I want to talk a little bit about ionic conductivity because my title was Ballet of Electrons and Ions, so I need to talk about ionic conductivity as well, and I want to show you results that might interest you as well. And actually, ionic conductivity, it's very interesting and can occur in polymer in a very easy way. And I like to do all this analogy. And here is a washing brush that everyone has in their kitchen. So when you return from work after a long day, you go and wash your dish, and this brush is very dry, stiff, and very thin, right? So if you take a jar of water and just pour on the top of this washing brush, the washing brush sort of absorbs the water and swells and becomes, no longer thinner, it becomes thick, and with a lot of water. And actually, if you look into the morphology of those polymers, you take a microscope and look into this morphology, you would see, something very similar to a plate of spaghetti, where the spaghetti is the molecules, is the chain themselves, and you can see there is a lot of porous entrances where, not only oxygen, but also, some sort of solvent could actually penetrate in. And this is basically how we actually active ionic conduction in those material. By using our ideal condition and solvent, we can hydrate or swell that particular polymer and all these small little pores in between all those spaghettis. And because you swell with solvent, a hydrate ions can also penetrate in and interact with the macromolecule, in the volume of the macromolecule. And remember that ions have charge, and by interacting with the macromolecule, it can dope the material. And this is exactly what happened. And ideally, you want to have a device to control this ionic conductivity that has 3 electrodes, and therefore mostly of this ionic/electronic conductivity are addressed by the use of transistor. And this device became very famous, and nowadays people call organic electrochemical transistor, or OECTs. And the reason and the structure is basically the one that I'm showing here, where you have 3 electrodes as every transistor, the gate electrode that actually is in contact with a electrolyte, and the channel of the transistor that is actually limited by the source and drain. And the difference from the traditional transistor is because instead of an insulating material, now you have an electrolyte, and an electrolyte has a solvent plus ionic species. And the way, the mechanism of conductivity or mixed conductivity ion-electron occurs on these transistors is summarized here. So in the off state, and by off state is the absence of gate voltage, the current between the drain and source is basically 0 because the semiconductor is initially neutral and there is no charge. And if you apply a voltage between source and drain with very little charge, sigma is very small, and therefore conductivity and current is very small as well. But as you go to the on state, and here I'm applying a negative bias to the gate voltage in related to the source, I push anions into the material. And to maintain neutrality of the semiconductor within the channel, the drain of the material injects 2 holes or extract 2 electrons, right, to maintain neutrality. And by injecting those charges to compensate the ionic species that is entering into the semiconductor, you increase the amount of charge, and therefore you increase conductivity, and therefore you increase current that you measure across the channel. So it's very easy to see how doping or how the movement of ions into the polymer activates or deactivates the conductivity of the channel. And by looking to this picture, you realize how easy within this structure of material, of devices, is to transduce or to translate ionic flux into electronic current, which is the technology that we dominate, referring to the very first slide that I mentioned in my presentation. And we call this electrochemical transistors because this interaction ion polymer is basically an electrochemical reaction. You have the neutral polymer plus, here a anion plus a hole, and then you create, a p-doped here, a positive doped polymer, and then compensated by this particular ionic species that actually entered the material. And again, those ions, they enter the bulk of the material. And therefore, there is a lot of space to receive charges. And therefore, the capacitance of these devices are huge. And by having a huge capacitance, you also have very, very big transconductance, which is basically the on-off ratio. The transconductance is basically the on-off ratio. And by this chart here, you can see that OACTs, here is P.PSS is another very important material in the field, are one of the highest by far transconductance transistor among several technologies. And by having a very big on-off ratio, you also have a very interesting or a very high resolution for transducing ion to electron fluxes. So there is a number of applications. I'm going to fly a lot faster here. I can, later I'm going to show my email. If you have any doubt, you can email me. But this is a work that we did with Roisin Owens from Cambridge. It's a biosensor. So basically the OACT is here. And it's very interesting because you can functionalize those OACTs with biological cells. And Roisin is basically a biomedical research. So she works a lot of this disease and is interested in bacteria and virus. So she wanted to do a biosensor for salmonella. And it turns out that is really fascinating the way salmonella acts in our body. I didn't know, I am a physicist, but when you interact with those people, you learn a lot. And basically, you have these epithelial cells in your organs, especially in your stomach. And those epithelial cells, they have this tight junction, is some sort of proteins that binds, two epithelial cell together. And what the salmonella does, which is a bacteria, is basically eats down all these proteins. So they sort of open up the gap between two subsequent epithelial cell. And if you think about your stomach, very acidic, now that gastric juice, which is very acidic, can actually go into your bloodstream, causing all this phenomena that everybody knows when you have salmonella in your body. So Roisin wanted to do a very simple biosensor to sort of measure if a certain milk would have or not salmonella. So what we did was very simple. We built an OACT essentially the way I just explained it to you. And on the top of the channel, we actually Roisin actually did grow the epithelial cell. And when the tight junction is actually there and holding the cells together, the ions cannot penetrate across the cells. And therefore, it doesn't actually get into the OACT. And therefore, you have a very small sort of current or doping process of the OACTs. But if you have salmonella, the salmonella will eat down, the tight junction, and therefore ions can actually penetrate among the cells and can give a very high current signal. So a very simple concept can lead, to a very good impact because it's not very good to drink milk with salmonella. So the idea here is to sort of have those small little biosensor in the milky box, and before buying it, you just break it, and then you see if something flashes or if something turns blue or green. Then you see, oh, this milk is not very good. Let me take another one. By just simply using, biology and physics and devices. And you can do a lot of things. Here is a work by Rivenay from Northwestern University. He actually sort of built devices similar to an electroencephalogram and compare it to the state-of-the-art equipment. So you can see the resolution is actually very big. And this is actually thanks to the very big capacitance of this transistor. And as polymer can be made flexible, you can actually use flexible substrate, and then you can fold. This can be into your body, can twist, and everything would work because polymers are compatible with flexible electronics. I think my time is about to finish. George, how much time do I have?
5 more minutes.
5. So
yeah.
I'm gonna just quickly talk about another work here, but before, I just wanna discuss quickly this literature map, because I think I'm talking to an audience of students, and I think this is very important. So when I did my postdoc at Stanford, I had a project which was bioelectronics, is basically what I have discussed with you so far. And then I wanted to do something impactful on the field. So the first thing that I thought, okay, let's do a literature map to see what people are doing and to sort of spot blank spaces in my field that I can actually contribute towards, right? So I understood that, there was a lot of biosensing, of course, there is, medical application in biosensing. So a lot of people is doing this. So the field was very crowded in biosensing. But I noticed two actually empty spots that are actually very important. I noticed that little to negligible work on fundamentals and really understand this transduction between ion and electron. And mostly of the application was actually gravitating towards, biosensor and maybe batteries and biointerfaces, nothing else. So I thought, okay, I'm gonna try to fulfill these two gaps, because of course, when you do something new, you sort of stands out, right? So one of the first contribution that I did was modeling OACT. So we developed a model. I'm not going to details, you can see, all these papers, but basically, we did device physics along with circuitry analogy, not only for the OACT itself, but the functionalized OACT as well. And by using, some mathematical tricks named Laplacian transform, we're able to, really recreate all these transient responses of OACTs, and even model biofunctionalized OACT or biosensors. And by playing with area and device architecture, we were able to sort of teach the literature to work on the optimal region of biosensors, which is not so clear or straightforward. But when you do modeling, you learn so much that you can understand that in terms of membrane resistance, there is an optimal operation region where you sort of enhance the resolution of your biosensor. So this is mostly what we learn here. And in terms of new application, I'm going to also fly very quickly. We actually developed something that we are very proud of, which is the neuromorphic devices. And neuromorphic devices are very interesting devices because it sort of goes towards what people call about neuromorphic computing or brain-inspired computing, right? And the reason people are looking into this type of architecture is because the von Neumann architecture is about is in a crossroad. If you think about the Moore law, we are duplicating, transistor every 2 years and etc. But if you look at the clock speed of the computing, because of several intricacies of electronics, the clock speed is not actually growing linearly as it was before. So although we are very good in doing photolithography and actually reducing the sizes of transistors, we are not harvesting clock speed on that matter. So people are, of course, doing a lot of tricks to enhance clock speed and etc. But, there is a discussion that maybe we are about to end the von Neumann architecture, period or dominancy. And one of the architecture that people are looking into is the brain-inspired computer. So it's basically to create computers that uses the architecture of our brain. And IBM is working on this, Google and a lot of big tech companies are interested on that technology. And the way you mimic the brain is to start understanding the brain. And I'm going to jump quickly, but two things that are very interesting in our brain is that our brain is not digital. We are actually analogic. So we have, a lot of number of memory states. It's very interesting. And our on-off ratio is not very big. It's around 4. So if the neuron potentiates or not, the intraneural region is about 1 to 4, this on-off ratio. So the brain is very good in resolution. And especially one thing that we really want to learn for the brain is what we call plasticity, how the brain learns, and then kind of bring this to an artificial device and going towards artificial intelligence. And when I was studying this, and I'm going to summarize quickly, I came across this multi-store model. This is very fascinating. I could actually speak another hour about it. But this is basically a paper long time ago, 1968, 1 year before we went to the moon. So it's a long time ago, by two researchers that were trying to understand how the brain holds memory. And this has a lot to do with rehearsal, how much you practice. And if you rehearse little, your potentiation, your memorization decays and goes to 0. But if you always sort of rehearsal and practice, your potentiation in sections of your brain, you start going up until you get to this persistent memorization or long time memorization, where you really learn something and never forget. It's interesting because you learn how to ride a bike after rehearsing a lot of times, and then you can spend 10 years without riding a bike. As soon as you get a bike, you are good riding it. It's very interesting how, as soon as you get to this persistent or long time memorization, things actually is, kept in your brain for a very long time. But if you don't rehearse enough, they sort of fade away. So it's very intricate. But what we did were to propose a device that would sort of behave similarly to this multi-store model. And the way we did was basically join together two of these OACTs. And the idea here is mostly to create a battery. So you have one that here, I think it's mostly, this picture is better. One that actually is doped, so is p-doped, and the one that is neutral. So we have basically a battery. So when you bridge them together, the doped electrode sort of sends charges to the undoped, sort of getting to the equilibrium. So you sort of change the conductivity of both electrodes because you are moving charges in and out. So if I do controlled discharge, I sort of create different memory levels. So I create some sort of analogic memory, which is the first part of a brain, to be, some sort of analogic behavior. And this is done by simply charging or discharging a battery and measure the conductivity of one of the electrodes. So I discharge, so it moves to a slower conductivity, so I kept that level. So I charge, it moves to another level, and etc., etc. And interestingly, this device sort of behaves very similarly to the memorization model that I just mentioned quickly. So by rehearsal for a machine is a pulse to charge or discharge the battery. So if you rehearsal a little, nothing happened, your memorization stays what we call sensory memorization, which vanished very quickly. But as you keep growing, you start adding more and more memorization level. So you rehearse a lot, you start playing your guitar every day, so you get very good at it. And if you start rehearsing and practicing, so you get to this very end of the memorization level, which is the long time memorization. So you really learn how to operate the device in that way or to do that particular task. And while in the brain, as soon as you get to the persistent memorization, you never lose it. You always have that memorization. I gave you the example of riding a bike. In electronic device, you can also undo this. So you can apply what I call unrehearsal pulse, and then you can erase that sort of memorization, which is not able in a biological matter. Well, it can be done, but of course not in a straightforward way. And therefore, you can see that, the curves, they look very much alike. And this is just to show that, we have a bunch of states, and they are non-volatile states. They are very stable. As soon as I get into a state, it gets very stable. And to finalize, I know that I'm running very quickly because of time. I think I like to speak a lot, so that's the reason sometimes I get a little longer in my talks. But we also did in this particular system, we tested a very simple test, which is the Pavlov dog. And Pavlov dog is a very straightforward test for learning ability. And basically, the idea is that you show a food to a dog, and the dog sort of, salivates. And then the second act, you ring a bell, and the dog doesn't do anything. And then the third act, you show the food and ring a bell, and of course, the dog will salivate. And especially in this act, the dog will associate the bell to food. And the fourth act, you basically ring a bell and the dog will salivate. So basically is inducing some sort of learning into the very simple ability of plasticity. And we actually did that on the paper. The details is in this paper here, in both papers actually. So we put it basically 3 neurons, which is, and by neuron, I'm actually mentioned 3 of these devices together to create some sort of a small neural network hardware. One is for sight of food, one is for bell or ring, and the other is salivation, which is the output. And here is the electronic that we used. I'm not going to detail, but what I'm going to show you is the response. So when you do the sight of food, you see that the response is high. So the dog is salivating. When I probe the bell ring, which is basically, stimulating, one of the neurons, it doesn't show up. This is just a capacitive response, but you see that the response goes to 0. And then when you do both, because when you bell the ring and sight of food, you actually apply voltage in 2 of the neurons. And the way we connected the 2 neurons is by a relay. So I can explain it later if you are interested on. But you can see that the signal is high. And after that, by just probing the bell ring, or applying a voltage here, sorry, you have here, I am at home, sorry guys, because George knows that my computer at USP crashed, so I'm at home. But anyway,
that's okay. That's okay.
Someone, my daughter is crying, but I think it's okay. So the bell rings here, and then you can see that now the signal as salivation is high. So you can see that, by this very simple electronic structure associating different artificial neurons, the dog actually salivated. More details I can give you into the question section, or you can actually download this paper. We did it flexible, but I'm gonna go towards the acknowledgement. I know that everyone is tired. So this is the group, and I acknowledge all my guys. Gustavo is a former student at Uberlândia, so he's now a postdoc here. I want to thank all the agencies that helped financing all this research. Alberto Saleu, my advisor at Stanford, Roshin, which is the biomedical research at Cambridge that actually helped on the biosensing, and Heinz von Segen from TU Darmstadt, which was my advisor in my PhD as well. Thank you very much. Sorry about the time. And here's my email address. If you want to contact me, I'm going to be very glad to answer any further questions and share ideas with all of you. Thank you very much. And again, sorry for the 10 minutes.
No problem. Extra. Thank you very much. That was very, very nice. Very exciting work. So we are open to questions. Anyone? It could be written, and then I write them. I read them to Professor Faria. So yeah. I have a few questions.
Go ahead.
So right at the beginning, when you said that there is a conjugation when you torsion the polymer, is that you instead of having the sequence double, single, you get a repetition of double or a repetition of single, or because it's like you create a barrier, right, between the two
yeah
types.
Well, your question is interesting, because that can happen if I create a polaron, which is if I add a charge, I can actually break the conjugation with single or double. That is actually very possible. But when I was talking about break of conjugation, is basically a torsion in the backbone. That's it. And that in that particular phenomena, you don't create a polaron or bipolaron. Polaron and bipolaron are created by doping process, and therefore, by doing so, you can break the conjugation by doing exactly what you just said. By instead of having single and double, you have single and single or double and double, right? But are different aspects. One is physical torsion, and the other is electronic doping.
Electronic doping. So when the electronic doping is a lower energy effect, right? And the torsion is a much higher, because it's it's a macroscopic quantity of energy.
Yeah.
So I had a little bit. No, I understand that there is. It's probably related to the where you are counting the energies from. But in your HOMO and LUMO,
are all
higher
positive,
energy
right?
was below. The HOMO is 5 and the LUMO is 3. I'm sure that you are counting from a different place that I am thinking, right?
No, this is HOMO and LUMO, and all this Venn diagram energy are all negative, because the zero is in the infinity, right? So I apologize, because normally we only say, the moduli, but you are completely right. Think about everything being negative.
So you are in reality, you're zero
is in the infinity, right?
In the infinity, okay.
Because
okay.
basically
was...
those work function is defined as the work that I have to do to extract the electron from that particular level and bring it to infinity, right? So this is basically the definition. And because zero is in the infinity, everything else is negative, right? Completely right. And I'm sorry about that.
No problem. No problem. When I thought it was very interesting the modeling you did using circuits and so on and so forth. I understand that you are looking at the phenomenological theory, right?
Yeah.
Kind of a phenomenological theory, and you model using, you know that this device behaves like a capacitor, and so you just make a circuit and you do the modeling. That's very interesting. But is there any modeling of the material by itself using the FT or something like that, where these materials are relatively simple, right? They are not too complicated, but still, I think the FT is getting into everything in condensed matter. I am sure you know, right?
Sure, absolutely.
They became so good at calculating properties with a lot of accuracy. One place that we are still holding a flag against them is strongly correlated.
Yeah,
that's where it's a little island where they cannot enter, but they enter almost everywhere. Is there a modeling of these materials using the FT?
Yeah.
I'm sure there is.
Well, that's the point. I'm actually very interested in modeling. As I showed you, I think there is blank spaces in mapping the literature. And modeling is a very blank one, for instance. So we developed this one. This is very simple. It's little of device physics and circuitry analogy.
I like it very much. Very much.
Basically, the idea that you just mentioned. And recently, we just submitted one that is based on thermodynamics and electrochemistry. But I haven't seen anything towards FET, which would be very interesting. And the reason is this is actually a relatively new field. Well, doping polymers with ionic species are not very new, of course, but the bioelectronic field, which actually pushed this mixed conductors, are relatively new, no more than 12, 15 years. And actually is application-driven field. People are looking to biosensor, biomedical devices, and etc. And there is little to negligible folks doing fundamentals. So there is a space for looking into it. If you are interested, I'm very glad to later we can jump into a conversation more informal, and we can try to find synergies.
Sure. We have. I don't do the FT. The FT are our colleagues here, Professor Hiroki and Professor Thomas Schmidt. I don't know if you, maybe from Gustavo, you know of them. They do a lot of, and I believe that Hiroki did something with polymers at CNPen in Campinas.
Exactly.
They have a lot of research on that there. So another thing I wanted to ask, let me check here if anyone raised his or her hand or is asking anything. No. Well, this is a little bit out of place because I was writing the question here as you were talking. So what about the technology in Brazil in this area? Are there startups? Are you involved in any startup? Are you thinking about getting involved in startup?
Yeah, that's a good question. I was still involved in a startup, but when I was back at Stanford, and that's the reason, I sort of mentioned to you that I normally say that I did 2 postdocs in science and one in sort of in entrepreneurship.
Okay.
And well, here in Brazil, I think there is still some elements, and by elements I mean it's still, we need to still build the ecosystem of it, because when you look at Stanford, and this is a particular view, it's my opinion, it's not based on study and etc. It's my experience on experience. So I tried to understand what was happening on that valley to be so rich in, you know, in creating new technologies and new industries. And I understood that there is 3 very important verticals. One is the human resources, which is, you know, fulfilled by Stanford, by UC Berkeley, and, you know, a lot of great universities around. So this is one. Number 2 is, and it's actually very important, is money, is financial, right? And
it's expensive, right?
Exactly, it's very expensive, you know, to develop new technology. You expend a lot of money. And I can give you details of my experience just to show you how, you know, very complicated it can get for you to become, you know, or your company to become a company that can succeed. And so there is the second vertical, which is money, which is very important. And the third one is services. And this is actually quite important. So just to put in perspective what I meant about services, one is the product realization lab. So we had, you know, a lab, completely lab, for us to prototype, to test ideas, right? So this is very important. But when we started the company, there is a lot of legal intricacies to start a company, to when you raise money through, you know, investors, there is always a lot of differences or intricate policies and regulations. So you need to have, you know, some sort of backup in terms of lawyers and accountability and etc. But a startup doesn't have the money for that, right? So you have to push your technology. And in Brazil, I noticed that a lot of companies, they die because they don't have enough either money or support to get started. They have great idea, they have a great team, but they fall apart because, you know, there is little to non-existent support. And by that, I'm going to give you an example. So when we raised our first money, we raised through a angel group. We went to a very big law firm in the Valley. The name is Wilson Sonini Law Firm, and they represent Apple. Look, they represent Apple, right? And we went there because there was a startup event promoted by Wilson and Sonini. So we went to this very big building. You know, for those who know America, law firms are very rich in America. They have a very active judicial power in there. So law firms are very rich. So the building was spectacular. And the idea of that particular event was to invite startups to showcase their ideas and to pitch their product. And we did the pitch. We had a product, we have a startup, we did the pitch among other colleagues that actually did the same. And afterward, we have a little get together, networking, and etc. And some of those companies that actually went to this event were invited to return to this law firm. And basically they said, look, we like what you do. We think there is a potential to be a successful company. We know you guys don't have the money to pay for a law firm with this structure. So basically we are offering you a a deal where we are going to give you for free all the legal support that you guys need until you raise Series A, which is, certain amount of money. After that, you guys is going to start paying us. And the only thing we ask from you for all this free support is that you're going to sign an agreement that you're going to stay with us until you exit, until you sell your company. And of course, they are law firms. They want the business, right? So, but this is very interesting because we were actually supported, or we were, we had our law firm the same as Apple. we had the same. That was
for
fantastic.
No money.
For no money, exactly.
There was a contract. Sure, they are not.
Exactly.
They are not a charity, right? They are not a charity. So
they say, well, there is a potential. I'm going to work for free, 2 or 3 years until you guys.
They are investing.
Very interesting. And of course, we did the deal because it was zero cost for us. And of course, you want to be supported by Wilson Sonnini because they are very big. They know how to deal with tech companies and etc. So we did the deal, and everything was actually made by them for free. They incorporated us in Delaware. They did everything, the notes. We had international investors later on, and by investors, international investors, there was extra paperwork. When we had to do an FDA approval, they did for us, and everything for free. If you do the accountability of that, would be in the US and especially in the Valley, hundreds of thousands of dollars. And a startup cannot afford that. But they knew that they were actually investing on us, and that money would pay back at a certain point. So, and I think this vertical is very important. And this happened with us in law firms, also and folks from the retail that would push the product to retailers. They did exactly the same, because they know if they actually take 10 or 20 startups, 2 of them will be successful and will pay back everything and more money plus, right? So I sort of understood that, there is an ecosystem at the Valley, which is, again, human resources and very, very stimulated ones that they go to Stanford. They want to create an impact on the world. So when they are doing their PhD, they are always, in contact with, successful entrepreneurs, a lot of talks about it, a lot of startup events, a lot of people pushing, for you to think of outside of the box using the technology you dominate to create an impact. So you have the human resource. Then you have investors. They give you money because you have to, to prototype a product. You have to fly to China to create something. You have to hire market people. You have to have a presence in the social media. So you have to hire, someone to deal with your social media. You have to hire someone to create your website. So you need money. You have to leave. So you need money. So there is this vertical and services that also support, early stage startup. It's not charity, of course, but they do support, and is a very important support that you would never get if you would pay for that support. And here in Brazil, you go to a law firm and say, "Oh, I have a startup. Can you actually help me here?" They show you a 50,000 Brazilian reais bill, and of course you cannot pay that.
To start talking.
Exactly.
But where is the bottleneck? Is it the government or the private sector mentality? The private sector mentality is still, we are in the last century, we invest in soy, we invest in oil, we invest in this and that, but
commodities, yes.
High technology is too complicated. We don't want to get there because we don't understand, we don't know what's going on. and we can never catch up with the Americans and the Europeans. Is the mentality or what?
I think we have to be honest with this. I think there is a little of our mentality. To be honest, I did all my study in Brazil, and the first time I really got into entrepreneurship was when I was at Stanford. So I think we, university. Of course, I'm a physicist, right? So we do a lot of fundamental, but I think we have to understand.
Which is fundamental, right?
Exactly.
It's fundamental for a reason. Yeah, it has to be done.
Exactly, but fundamental, what I learned at Stanford is you only make an impact on application if you dominate fundamentals. Otherwise, it's trial and error, and you never get there, trial and error. So you only do an impact.
Someone will get there before you because they know the fundamentals.
Exactly. There we go. You statistically, it's gonna get further down the road, right? So you have to be very good on fundamental. This is a very good thing from Stanford. They really learn a lot of fundamentals, even the engineering persons. So, for instance, we do here classical mechanics that goes to Lagrange and Hamiltonian framework, right? The engineering folks at Stanford, they also learn that. And here we don't teach, classical mechanics to our engineers, which is very important, right? So I saw, small things that it says that, maybe the way we showcase how important fundamental is to, impact on application, it's not well bridged in Brazil. And I saw that, at least my experience, everything I'm saying on this answer, it's my experience, was actually very well seasoned at Stanford. So I think answering your question is there is a little bit about us that we have to improve and understand and be maybe more thirsty to impact on something. And of course, we have the problem with the industry as well. And I think the industry doesn't trust, our ability to develop technology on that matter. And I think that's actually very true because they prefer to import from the US, from China, from Europe, from, other countries instead of, risk investing here and developing our own technology. And this goes also, it goes all the way down to the, to the politics support. There is a lot of intricacies that makes, our productive sector afraid of, investing on our own technology. So, it's a very complicated subject because it's all correlated, right? And you can also mention another very important aspect is also our tax structure. It's very complicated. If you talk to a company here, normally they don't really know if they are actually paying the right taxes because it's so complicated that for a startup that doesn't have a good professional support, it actually can get a fine that would terminate the company forever.
I know cases like that the company was basically shut because they didn't understand what they had to do, and then they didn't know that they had to pay, and 2 years in a row with the fines and the interest, they are broken.
Yeah, there you go. Yeah. So I think we have to change a lot of different, sectors to become more comfortable to promote this entrepreneurship and startup. And the way we start with it is by talking about it, right? So it's very important to discuss and make pressure to more policies to become debated on the parliament and etc. I've participated into a round table in the last MRS in Brazil, where we have actually discussed that we physicists and material scientists and engineering, we have to create a community to support representatives in the parliament. Because, for instance, it's very sad. But do you know who was the head of the Innovation and Science Committee at the Senate a few years ago? Romário. Romário, the player, the football player. Nothing against Romário. I love Romário.
I admire him very much.
He doesn't understand science and technology, and he was the head of the committee. So where is, Vanderlei Baiato, Glaucius Oliva, all these people? They have to be there. A lot of people from all the federal universities, we have a lot of people that have great mentality and understands the problematic, and they have to be there. They have to represent. They have to say, no, this is wrong. Otherwise, you're going to kill this sector. We have to promote the sector. So we scientists are very bad in create representatives, I guess, and we have to be better on that, because that is the way we change policies. And look, we are having a very big problem in São Paulo as we speak. The Alesp, which is the parliament of the state parliament here, is approving a law that will reduce in 30% the budget of FAPESP. And
that's going to be a disaster. That's going to be a
disaster. It's crazy. And you go at Alesp, the parliament of state of São Paulo, and you look at the folks that are in that parliament. You don't have a single scientist, a single professor, or one or two professors, but not university professors. So we have to be better on that. And this is the way we change and impact. And I guess, the society has a bit of work on that. They can sort of start talking about that and stimulate good people to, become our representatives. I think Ricardo Galvão is sort of tracing that way. Hope, he can succeed. But we as a community, we have to be very strong and support those people that really want to make impact where policies are decided. This is really important as well. So there is a lot of aspects, but
we can talk about that for hours.
Exactly.
Let me see if there is anyone. Please, if you have any questions, you can write on the chat and I can read them. Let me quickly, I have a few other questions here. I was the OECT, but I think that's not the point. I was going to ask how fast is it? As a transistor, but that's not the point, right? You are not trying to do
it's very slow.
To do something. It's very slow, right?
Yeah,
but that's
because capacitance is very high.
Yeah. You're not trying to build a computer. You are trying to operate in an environment which mixes liquids and ions and electricity and everything, where there is nothing else, right? How can you build a transistor in an environment like that? And that's a system. And I was amazed that the on-off ratio is quite high, right?
It's very high. Yeah,
it's very high. So
it's very high because basically we don't talk about. It's interesting. We talk about volumetric capacitance, and people doesn't like this, but basically it's because ions go into the volume of the material. So you actually swell a lot of charges, and therefore you promote a lot of doping or de-doping of your semiconductor. So you actually switch on and off in the conductivity in a very big differences. For instance, one of our devices has an on-off ratio of 10 to 6. It is very big.
Okay.
Yeah, it's
this
really
is
big.
Huge.
But it's slow, but it's very slow. You see, this one is
Yeah.
a big device. You operate in 20 seconds. Very big devices are very slow because there is a lot of space for charges to go in.
No, that's clear. Yeah, that would be the slowest computer in the universe, right? But that's not the idea. You don't want to use that to build a computer.
Exactly, but remember, the operating speed of our brain is not very fast. It's 10 hertz. It's 10 hertz for young people. Us is 1 hertz or even smaller.
1 hertz. Oh my goodness.
Yeah, but this is actually so. We always think about the von Neumann architecture. The brain architecture is different.
Yeah.
It operates differently,
right? Parallel. You are
exactly. Yeah. It's different. So we don't
probably
need
much more complicated than that. It's not only parallel. It's something.
No, yeah, it's very intricate, but it's we are starting getting into there, and this neuromorphic computing is actually pushing the understanding of the brain. So it's actually very interesting, the synergy, because we push biology, and biology help us to mimic, what happens in the brain. So it's a multidisciplinary area for sure.
Yeah, that's for sure. Okay, so I think we are way 105 minutes. That's
I apologize.
That means that it was interesting, and we have basically almost everybody is still here. So
great.
They didn't ask questions, but I am sure I'm going to ask you for the slides. I send you an email tomorrow. If there is anything there that you want to remove, just for... I will post together with the video that I recorded of your seminar. Sure. And you gave your email at the end, so I'm sure we will have students here who are going to be contacting you to ask questions and maybe look for opportunities at your lab. Who knows?
Sure. Yes, I'm here to answer questions and to talk about science for sure.
Okay. Great. So Gregório, thank you very much again.
My pleasure.
And send a hug to Luis Nunes if you see him.
Absolutely, I will do it. So.
Okay. Thank you very much.
See you then, and thank you for having me.
You are very welcome. Thank you.
Bye-bye.
Bye-bye.